DNA is very important in DNA replication. How do you get more DNA? So DNA is a double helix. Notice that you have a 5' and a 3', whereas on the other end of the chain, you have 3', 5'. So vice versa. Now, why is this? Remember, DNA is a double helix, but each chain runs anti-parallel to each other. And that's why you have a 5', which is carbon number 5, and 3', which is carbon number 3 of the 5-carbon sugar component. So nucleic acids have 3 components: the sugar, the phosphate group, and the nitrogenous base. So what are the steps? Steps 1, helicase, an enzyme, unwinds the DNA. Now, when that takes place, step 2, single strand binding proteins keep the DNA double helix separate. The most important enzyme, DNA polymerase, adds nucleotides to the new chain. So notice that DNA polymerase can only add nucleotides in the 5' to 3' direction. So the leading strand has a continuous new DNA strand. Now notice one DNA template is used, or the template strand is used, and then the other new strand is added. So this is an example of DNA being semi-conservative, meaning that an original strand was used. So continuous strand form in the leading strand. The other strand is called a lagging strand, and this is why. So remember, DNA polymerase can only add in the 5' to 3' direction. Well, this is opposite. So what must take place first? The enzyme primase actually forms a small RNA primer. Now, as this RNA primer is formed, you now have the DNA polymerase forming or adding new nucleotides to this primer. Now, it cannot add continuous. It actually forms these short Okazaki fragments. So, how do you connect these Okazaki fragments? Well, we use this DNA ligase, which attaches these Okazaki fragments to now become a continuous strand. Now, this takes place in the lagging strand, DNA replication.